A step towards cloning humans? Chinese monkeys become the first primates in the world to be replicated using the Dolly the Sheep method of transferring DNA. Identical macaques Zhong and Hui were born 8 and 6 weeks ago. The monkeys are the world's first primates to be cloned from transferred DNA. Study marks a watershed in cloning research and raises major ethical questions. Scientists hope to pave the way for populations of genetically uniform monkeys. Groups could be customized for groundbreaking research into human diseases. Scientists yesterday revealed they had cloned 2 monkeys, potentially paving the way to copying humans. Using the technique that led to Dolly the Sheep, a Chinese laboratory has created a pair of identical macaques. The researchers said they had cleared the final barrier to human cloning and offered hope of treatments for Alzheimer's, Parkinson's and other illnesses. The monkeys are the most human-like animals yet to be created through true cloning, effectively carbon copying the DNA of a single individual. 2 monkeys have followed in the footsteps of Dolly the Sheep by becoming the world's first primates to be cloned from transferred DNA. Identical long-tailed macaques Zhong, pictured, and Hui were born 8 and 6 weeks ago respectively at a laboratory in China. Mu-ming of the Shanghai research team, was asked yesterday whether the same method could be used on human cells. He replied, "Yes. A macaque monkey is a primate species, humans are primates, the technical barrier is now broken." But campaigners said the development was worrying. "We are concerned that this is a stepping stone to the creation of human clones," said Dr. David Qiang of the lobby group Human Genetics Alert. Although it looks like that would be technically difficult, those with enough financial resources and the ambition to be the first to create a cloned child are likely to try. The monkeys, Zhong and Hui, are the first primates to be cloned using the somatic cell nuclear transfer technique. DNA was taken from a macaque fetus cell, put in an egg and fertilized artificially to form an embryo later born to a surrogate mother. The same method was used more than 20 years ago in Edinburgh to create Dolly from a single adult udder cell. Cats, dogs, rats and cows are among 23 species of mammal also to have been cloned in this way. Zhong and Hui were born 8 and 6 weeks ago respectively at the neuroscience arm of the Chinese Academy of Sciences. It took 21 surrogate monkeys, which had only 6 successful pregnancies, for the 2 cloned monkeys to be born. Another pair created from the DNA of a live adult failed to survive. The success is credited to reprogramming the genes of the cloned embryos to allow them to develop properly in the womb. DNA taken from their ears confirmed that Zhong and Hui were completely identical. The 2 females are being bottle-fed and are said to be growing normally. Dr. Pu said his team had no intention of using their method to create humans. However, Darren Griffin, professor of genetics at the University of Kent, said, "The first report of cloning of a non-human primate will undoubtedly raise a series of ethical concerns, with critics evoking the slippery slope argument of this being one step closer to human cloning." Technically the first cloned primate was Tetra, a rhesus monkey born in 1999 through the much simpler method of embryo splitting. However, experts say this method, which replicates the embryo splitting to form twins in the womb, is not really cloning but just enforced twinning. The latest DNA method, described in the journal Cell, allows limitless clones instead of a maximum of 4. Professor Griffin said, "A primate model that can be generated with a known and uniform genetic background would undoubtedly be very useful in the study, understanding and ultimately treatment, of human diseases, especially those with a genetic element." Careful consideration now needs to be given to the ethical framework under which such experiments can, and should, operate. Professor Robin Lovell-Badge, a cloning expert at the Francis Crick Institute in London, said he did not believe the research increased the chances of humans being cloned. "The work in this paper is not a stepping stone to establishing methods for obtaining live-born human clones," he said. "This clearly remains a very foolish thing to attempt. It would be far too inefficient, far too unsafe, and it is also pointless." Cloning has captured the imagination of science fiction writers, with human clones created for spare organs in the film and novel Never Let Me Go. Dolly was cloned at the Roslin Institute in 1996 and named after country singer Dolly Parton. On the new experiment, Dr. Julia Baines, Science Policy Advisor at PETA UK, said, "Experimenters constantly receive funds to perform monstrous experiments on animals, and cloning monkeys is the latest frankincense that PETA condemns. Cloning is a horror show, a waste of lives, time, and money, and the suffering that such experiments cause is unimaginable. Because cloning has a failure rate of at least 90%, these two monkeys represent misery and death on an enormous scale."